Well, I would like to thank you, organizer, for giving me the opportunity to present our work on inflammation-mediated mitochondrial transfer from immune to non-immune cells. I would like to thank my lab member, my boss, Dr. Leon, and funding from NIH and research. We know that mitochondria has a life cycle of fusion and fission. So mitochondrial life cycle begins with the growth of pre-existing healthy mitochondria by biogenesis and with the removal of unhealthy mitochondria by autophagy, between which mitochondria continuously undergo fusion and fission. We know that there are three mechanisms of mitochondrial transfer: intercellular or horizontal transfer. First is with the tunneling nanotubes, through which mitochondria move from cell to one cell to another cell. Then second is the ejection of mitochondria in extracellular vesicle containing mitochondria for delivery to recipient cells. And third one is to release of free mitochondria for capture of by recipient cells. So how inflammation influences the mitochondrial transfer? So inflammation is your body's alarm or hearing system, essentially when controlled, and harmful when it is prolonged. So liposaccharide is a key player in systemic inflammation and has a pathogenic associated molecular pattern based pro-inflammatory effects. Upon activation, macrophage has a metabolic shift from oxidative phosphorylation to aerobic glycolysis, which is Warburg effect. Takes place in that, and mitochondrial ROS production increases and break in the TC cycle and accumulation of metabolic succinate. And succinate is the inflammatory signal, and it stabilizes HIF1 alpha, which drives the interleukin 1 beta production. And mitochondria becomes fragmented or produce more ROS, which also triggers inflammation like NLRP3. However, uncontrolled excessive mitochondrial fusion can lead to pathological mitochondrial fragmentation associated with the mitochondrial dysfunction and ROS production, and it impairs the organelle's ability to meet cellular demands and contribute disease progression. So this is the path why it shows the pro-inflammatory response. So how inflammation influences the mitochondrial transfer? To check that, we have done some experiment mechanism of communication through mitochondrial transfer during inflammation, and correlation between the mitochondrial bioenergetics and dynamics and mitochondrial transfer we want to check. So our hypothesis is inflammation alters rapidly mitochondrial dynamics in macrophage, packaging fragmented mitochondria into vesicles for transfer to neighboring cells, non-immune cells, potentially as a protective mechanism against the inflammatory damage. So we use we did in vitro experiment. In this we use a peritoneal macrophage and brain microvascular endothelial cells. Peritoneal macrophage residing in the peritoneal cavity plays a crucial role in immune surveillance, controlling infections and maintaining tissue homeostasis, acting as the first line of defense against the pathogen and controlling the wound repair and inflammation resolution. And non-immune cells like brain microvascular endothelial cells, which forms a blood-brain barrier in response to inflammation by releasing cytokine and chemokine to recruit immune cells to the inflammation. We use this mice strain, which expresses mito-T2. So the first question: what changes occur to peritoneal macrophage upon LPS treatment in culture condition? So we have seen that in the control to compared to LPS treated mice, we can see that the number of mito per cells get decreased. At the same time, the volume of mitochondrial volume per cell is also getting decreased. Here we are showing that peritoneal macrophage mitochondrial morphology. We can see the in LPS treated that the number of mitochondria get decreased, and in the others graph mitochondrial network analysis, you can see the density or footprint of the mitochondria also decreases, as well as the length of the mitochondria is also decreased. So, does mitochondria transfer to brain macrovascular endothelial from peritoneal macrophage upon LPS treatment in co-culture condition? So here we can see that in the control condition, the red one is the host mitochondria of the endothelial cell, and upon treatment we can see that there is a transfer of mitochondria from macrophage to endothelial cells. And we can see here that percentage of mito transfer in the cells which is very high. So mitochondrial transfer from macrophage to endothelial cell. Here we can see that white is the host mitochondria, and the green, which is the mitochondria which are transferred from the macrophage to the endothelial cell. So in summary, inflammation will not only disrupt the mitochondrial dynamics but also enhance the mitochondrial transfer between immune and non-immune cells. However, it remains unclear whether these processes are mechanistically linked or occur independently. So further, we have to investigate the molecular underpinning of inflammation-induced mitochondrial transfer and its role in intercellular communication. Thank you.
I think that was very quick, well done. We have time for a couple questions, if anybody has any.
I was wondering if you analyze the network, you use projections or, sorry?
Yeah, we use microscope, confocal.
Yeah, but do you use Z-stack, maximum intensity projection, so what do you do? Because obviously your networks
my colleague, Dr. Velu will show in his presentation how exactly it is getting inside the cell. Yeah, it is not just on the top of the cell.
Questions for the other at the back?
Actually, this question is from the previous speakers that were there, but I don't know exactly that. Maybe it is Andreas Casino. Yeah. So the question is like, and other also. The question is there any other biomarkers that we can? Because we have seen a lot of presentations where. On the cellular level. So is there any biomarkers other than ATP which can be used for, and
so you're thinking about any other biomarkers that can be used to assess mitochondrial state or function after isolation? Oh, well, you have, for example, different ones that can help you to measure the membrane potential. In a way, but for all those, you need light microscopy, and when you need to apply them quickly, you need to find the ones that are the most weakest. So you have electron microscopy if you want to take the time in order to isolate it and to see the stability. Then you have the ATP production when you isolate it and you measure it in orovaral system or in seahorse. Then sometimes you could use GC1, but sometimes you have to do a lab setup in order to prevent it from not having a clear signal. And then in order to, for example, for me, the easiest way is to assess, for example, if you transfer mitochondria with fresh mitochondria in comparison to other types, one of the things that we do is to measure the outcome. For example, if we have the same proliferation status or something else.
Thank you so much.